I'm Armelle Cuvillier. I founded B-Cell Design with Gaël Champier in 2007 to support us a board of directors truly committed to the development of our activity. They all share solid expertise in drug development. They all held key positions in pharmaceutical industry, and Dr. Jean-Yves Blay, the director of Léon Bérard Institute, heads our scientific board. The innovation of B-cell design is based on a genetically modified mouse platform to produce the four major isotypes of antibodies, but separately in each transgenic mouse model and directly with human characteristics. The strength of the platform is to generate a very large number of specific monoclonal antibodies by a simple immunization of the right mouse line with any kind of antigens from infectious agents, cancer biomarkers, cytokines, or growth factors. IgA is the antibody of the mucosa. We have chosen for the proof of the therapeutic concept of IgA the first more relevant indication, which is for us the metastatic colorectal cancer. Colorectal cancer is the third most commonly diagnosed cancer, and its burden is expected to increase. In France, more than 150 patients learn every day that they have a colorectal cancer, and more than 15,000 will die every year. In terms of available treatment, only 30% of patients will respond to the first-line chemotherapy, and of those who remained, half have a mutated KRAS gene, so won't receive an anti-EGF receptor immunotherapy. And finally, less than a quarter will show a significant cancer mass reduction. So what do we have to offer to those who stay? We are developing a new concept of IgA-based immunotherapy with new features that have never been used to date. Our first drug candidate is a monoclonal IgA anti-CEA, a biomarker overexpressed by 95% of colorectal cancer cells. On a late metastatic model of colorectal cancer, the anti-CEA IgA demonstrates a significant higher therapeutic benefit on tumor size reduction than the immunotherapy gold standard, the IgG anti-EGF receptor or cetuximab. On a chemoimmunotherapy combined treatment protocol, the orally administered anti-CEA IgA reduces the tumor burden, while the anti-EGF receptor IgG, the therapeutic, failed to control tumor growth. This is the first demonstration of the efficacy of an oral immunotherapy to treat solid tumors. The 3 years Gantt plans to bring out drug candidates in phase 2 clinical trials in 2023. Prior to that, we need to perform preclinical toxicity and safety studies to validate the GMP batches of oral formulated IgA, for which I am currently seeking funding. Thank you for your attention.